Well, welcome everybody to beautiful, rainy, cloudy, cold Champaign-Urbana, Illinois. I hear it is snowing up in Toronto, but I still would love to visit that city. I haven't been there a long time. Maybe Chris can get me an invitation up there for an in-person talk, eh?
We'll work on
So I'm going to come at exercise from a different angle, and that's from your microbes. So about 10 years ago, we got involved in a study looking at ulcerative colitis, and that drew us into into microbiomania. Actually, my students brought me into this, and so it's been quite a journey. So today I'm going to talk about microbes and exercise, and at the end a little bit about how they might be involved in endurance training adaptations. So what is your gut microbiota, also known as your flora? So these are the microbes, the bacteria, archaea, viruses, fungi, and bacteriophages that live in and on you. And there's actually more genetic material in cells of your flora than there are of you. So it's probably logical that they're going to have a powerful impact on you. These microbes could be commensal. They could kind of be benign, I guess. They could be there and not have any harmful coexistence. They can be mutualistic or symbiotic, so they could benefit from you and you could benefit from them. Or they could be opportunistic and turn into pathogens and take advantage of you and for their own good. Microbes live in a very competitive environment, especially in the gut. There are a lot of microbial niches and battles between families of bacteria on maybe the mucosal layer of your gut or in the lumen. And really their success depends on the environment they reside in and the resources that influence, how they operate. And certainly exercise might alter those resources. And so I'm going to show you some data today. You probably know that each person's biota is unique. Each one of us has about 500 species of bacteria living in our large intestine. Kind of like your fingerprint, your microbiota is unique to you. There's different types and species present in different people, and their relative abundance can be different. So even if you and I have the same bacteria, there could be very different amounts. After a few years of life, your microbiota is pretty stable in your gut. It can be knocked out of whack. That's known as dysbiosis by stressful events or changes in diet or other things, diseases. And it's really, because each person's biota is unique, it's really hard to compare between individuals. So the best studies tend to be ones that are repeated measure studies. You also have to remember that you have a skin, oral, and vaginal biota too. So it's not just about your gut, which we of course sample by taking fecal samples, but also there's studies going on looking at skin, oral, and vaginal biotas. So what do gut microbes do? Well, within the gut, they compete for resources and survival. Like I said, they actually can, some strains can actually feed on your mucus layer. That's interesting. They help us absorb energy. A lot of bugs can ferment insoluble fiber that gets passed through the gut tract down into the later regions of the small and the early regions of the large intestine. And those insoluble fibers can be used for fuel by these bacteria. And they generate metabolites like short chain fatty acids that actually can have effects on the host. So these microbes produce metabolites, enzymes, hormones. They detoxify some toxins, and they can affect your gut barrier in a good way or a bad way. They also communicate to our bodies. They promote maturation and development of the immune system. They promote or inhibit inflammation. There's a direct link from the metabolites of the gut microbiota to the liver through the hepatic portal circulation. They communicate through the gut-brain axis. You may have been reading literature these days that maybe microbiome is related to anxiety and depression or Parkinson's disease. There's certainly been some relationships. Importantly for this talk, there's some research that suggests there's communication of microbial metabolites through the gut muscle axis, which could impact exercise. And they also regulate metabolism, which could also impact exercise. And we're really just beginning to understand the role of gut microbes in human physiology. So what's the good news? Well, the good news is what you do can influence your microbiota. That is both its beauty and its potential. It's also why there's so much interest in it right now. If we can find ways to harness its beneficial powers, we might be able to improve health. As you might suspect, certain environmental factors shown here on this slide shape your microbial community, good or bad, whether you're breastfed or bottle fed as an infant, your environment, the drugs you take, stress. Your diet has an especially strong influence on your gut microbes, as you would predict. Importantly for this talk, though, what about exercise? Can exercise influence your gut microbiota? And if so, does it contribute to exercise-induced adaptations? Today, I'm going to tell you about several studies that we've done over the last 6 to 8 years that kind of looks at this. So to truly understand gut microbiota, we have to answer these questions. We have to answer who's there, what are the microbes or the bacteria residing in the gut, and do they change? What are they doing? In other words, what are they producing? What metabolites? What are the effects of those metabolites on the host? And what are the mechanisms of communication? So for who's there, we've leveraged the recent advent of next generation sequencing, DNA sequencing, to look at families of bacteria and other microbes in samples taken from humans and animals. Typically, one analysis analyzes 16S rRNA, which is common to bacteria. We could also use metagenomic sequencing to look at all of the microbes that exist within the sample. You may have heard the term gut microbiome, and really gut microbiome is the genetic makeup of the biota. So when we do this sequencing, we call it gut microbiome analysis. For what they're doing, we use metabolomics, either untargeted or targeted. To understand how metabolites affect the host, we manipulate those metabolites or the microbiota. Probiotics would be one example of manipulating microbes. Regarding the mechanisms, these metabolites can communicate with us in many different ways. They can diffuse. They can act on specific receptors. They can act epigenetically. They can act through the vagus nerve, the sensory nerve of the vagus. And also they can act as hormones. So I think it's safe to say now that exercise training does shift gut microbiomes in animals. And you can see some of the titles of a small selection of work that has been done in mice and mainly mice, but also in some other animals. To give you one example from a former student of mine, Jacob Allen, who's now actually an assistant professor here at Illinois, you can see the title I've highlighted here: Voluntary and Forced Exercise Differentially Affect Gut Microbiome in C57 Mice. So Jacob used phylogenetic sequencing information and principal components analysis to compare and identify factors explaining differences among microbial communities in either fecal samples or cecal samples. This type of analysis is called beta diversity analysis. So this graph shown here helps us compare microbial communities amongst individuals, in this case, in individual mice. Simply put, each dot represents an individual's bacterial community, or a person, or their personal zoo of microbes, if you will. Dots that are closer together are more similar. Dots that are far apart are more different. So it's really a big picture type of analysis. We compared the gut microbiomes of 3 groups of mice: mice that were sedentary, shown in red; mice that were forced to run on a treadmill, shown in blue; and mice that had free access to a running wheel, shown in orange. We compared these 2 types of exercise, voluntary or forced, because they are the best and most common ways in which we study endurance exercise in mice. And what you can see here is that the 3 groups clustered together differently. Bottom line from these data was that the 2 types of exercise caused the gut microbiomes to be different from one another, and both types of exercise were different from the sedentary control group microbiome. And this difference wasn't due to diet, because the mice ate the exact same diet throughout the study. So what about exercise in the human gut microbiome? You can see some of the titles here in this graph, and if you look closely at them, you'll notice something. So one of them down here says the microbiome of professional athletes differs from that of more sedentary subjects in composition, particularly at the functional metabolic level. What's common to all these studies is that they're cross-sectional, and we can't make any cause and effect conclusions. Some have failed to control adequately for diet or antibiotic use. Certainly, if you're comparing professional athletes with sedentary people, there could be big differences in diet that could explain microbiome differences. Remember, there's high inter-individual variability in gut microbiomes, making between-group comparisons difficult. So clearly, what was needed was a repeated measures longitudinal exercise trial. And so with that, Jacob, shown here on the left, and my other former doctoral student, Lucy, spent about 2 years of their young lives, doing a study we called FitGut. And really the aim was to understand whether 6 weeks of endurance exercise could change the microbiomes of previously sedentary lean and obese adults. We included obese adults because obesity is associated with dysbiosis and also a higher incidence of problems in the gut like inflammatory bowel disease or IBS. So we recruited 32 sedentary, lean, and obese adults and asked them to perform supervised moderate endurance training exercise for 3 days a week for 6 weeks. In addition to other measures, we collected fecal samples for microbiome analysis before and after the interventions, shown as E0 and E6 here. Importantly, we controlled for the influence of diet by having them eat the same meals before every fecal sample for a couple days. We did this so we could attribute changes in the gut microbiome to exercise and not to any changes in diet. Importantly, to further convince ourselves that exercise or sedentary behavior was changing the fecal microbiome, we included a 6-week washout period after the exercise intervention, hypothesizing that any exercise training-induced changes would revert upon reintroduction of sedentary behavior. So we took that sample 6 weeks, the W6 time point. So using the same type of beta diversity principal components analysis as before, we compared these people before and after exercise. Remember, dots closer together are more similar, dots farther apart are different. Obese subjects are shown in blue, lean subjects are shown in red. At baseline, note that there was a large difference in the gut microbiomes between lean and obese. As I mentioned, this was not novel, as other studies have demonstrated this. Obese people tend to be dysbiotic when compared to lean, different from lean. What was interesting and novel was that after 6 weeks of endurance exercise, the gut microbiomes of the lean and obese people were not significantly different from one another, as shown here. They were different to begin with, and then exercise made them more similar. However, just because they appeared more similar didn't mean they were exactly the same, as I'll show you in a minute. The two groups changed orthogonally, meaning that the same bacterial taxa that changed in response to exercise in the lean group were different than in the obese group. But the overall community structure, as measured by this beta diversity analysis, appeared more the same. When we looked at the week 6 post-washout, where they returned to sedentary behavior, this was also evident. The groups were not significantly different, indicating that exercise-induced changes in the gut microbiome were rather long-lasting, at least up to 6 weeks in this study. So we concluded that lean were different from obese, that exercise led to orthogonal changes in the gut microbial community structure, and that these changes that occurred in washout were orthogonal as well. So for all you microbiologists out there, here's a list of the bacteria that changed in response to exercise. Bacteria that went up are shown in green. Those that decreased are shown in orange. What's more interesting than their names, though, is what they do. Importantly, exercise increased the number of bacteria that produce short-chain fatty acids, more so in the lean people than the obese, as you'll see in a minute. Also, exercise decreased some harmful bacteria in the obese participants, Agarthella and Doria, bacteria associated with gut diseases. Given that we saw increases in bacterial families that had the potential to produce short-chain fatty acids, we actually measured short-chain fatty acids in the participant fecal samples. Note here that many of the effects of short-chain fatty acids on the host are also effects induced by endurance training, perhaps implicating exercise-induced short-chain fatty acids as mediators of some of the beneficial effects of exercise. So here you can see graphs that depict acetate, propionate, and butyrate, 2, 3, and 4 carbon short-chain fatty acids. The y-axis is micromoles per gram of dry matter base, so we have to remove the water content from the feces because that can confound the short-chain fatty acid measurements. And what you can see here is that the lean group, after the exercise intervention, had significant increases in acetate, propionate, and butyrate, the short-chain fatty acids. There was increases in the obese group, but not nearly to the extent that the lean group demonstrated. When we look at the samples in the washout period after the 6 weeks of return to sedentary behavior, in the case of propionate and butyrate, these levels came back down, indicating that short-chain fatty acid effects were kind of transient. So the take-home messages so far is that endurance exercise affects the microbiome and its metabolome in mice and humans. And so, of course, we want to address other questions. like, do exercise-induced changes in the microbiome and metabolome affect colon health? I mean, these bugs are right in contact with the colon. It makes sense that they would affect colon health. So we did some studies to look at that. And then also, are microbes important for endurance training adaptations? So in order to address these questions, we utilized two tools that allow researchers to study the influence of microbes on host physiology. We used antibiotics. So we gave the animals antibiotics in their drinking water, actually broad spectrum antibiotics. They drink the water readily, and the antibiotics gets into their system and into their gut. And we also used germ-free animals. So these are very special mice, very expensive mice that are raised in a sterile environment and have never seen a microbe in their lives. They actually kind of live in a bubble, which makes them difficult to work with. So there's pros and cons to each of these two tools. For example, antibiotic treatment doesn't completely kill all bacteria or affect other microorganisms like viruses. There may be also some side effects of antibiotics, but as you know, these side effects are pretty benign. If you've ever been on antibiotics, probably haven't had too many problems. Probably the biggest limitation of germ-free mice is that they develop in a world without microbes, and this affects their maturation and development when compared to animals that grow up in the world of microbes. For example, germ-free mice are actually hyper-responsive to stress and exhibit exaggerated hypothalamic pituitary adrenal axis responses. So using germ-free mice, we addressed whether exercise-induced changes in the gut microbiota could affect colon health. You can see from the title here on this slide, kind of gives the punchline. Exercise training induced modification of the gut microbiota persists after microbiota colonization and attenuates the response to chemically induced colitis in gnotobiotic mice. So what did we do? In collaboration with Mayo Clinic, we transplanted gut microbes from sedentary and exercise donor mice into germ-free recipient mice to determine whether they could affect colon health, both in the absence and presence of a chemical that causes ulcerative colitis. That chemical is dextrans sulfate sodium. Importantly, these germ-free recipients did not exercise. So what we were trying to do was to transplant the beneficial effects of exercise just by transplanting the microbes. What did we find? Well, I don't have time to show you the data. If we have time at the end, I can show you a few slides. But when compared to the sedentary mice transplant, the exercise mice transplant had overall healthier colons, reduced colon inflammation, a potential for improved gut barrier function because there was more mucus observed in their goblet cells upon histology. There was reduced symptoms in response to the experimental colitis treatment. And we saw increased healing signals after colitis. There was a higher IL-22 and TGF gene expression in their colons. So the next question is, are microbes important for endurance training adaptations? So these data are actually unpublished, some of which really has just been generated in the last few weeks. And I haven't had a lot of time to interpret it. So I'm hoping that some of the muscle physiologists in the crowd can give me some direction and help. So we hypothesized that antibiotic-treated and or germ-free mice would exhibit reduced adaptations to voluntary wheel running. That is, if microbes are important in the adaptations to exercise, knocking them down with antibiotics or using germ-free mice, we should see reduced adaptations. So the design for the antibiotic experiment is shown here. We use conventional specific pathogen-free C57 mice. We use 40 total, 10 per group, males and females. I don't have time to get into the sex differences today. We gave them half the animals antibiotics in their drinking water, and then we exposed them to telemetered running wheels for 6 weeks. We measured body weight, food and fluid intake intermittently throughout the 6-week period. We did an endurance test on the treadmill before and after the wheel running intervention to test performance and adaptation. And then we collected some tissues. And you can see the antibiotics that we used here. 2 of these are poorly absorbed, so they mainly affect the gut microbes, and ampicillin and one other are actually absorbed pretty well. In the germ-free animal experiment, we had 10 per group. There was a group that remained sedentary with a lock wheel, and then a group where we had a telemetered running wheel in the actual special cages. This really wasn't a trivial thing. We had to really work with our animal care staff and sterilizing these wheels and getting them tested and verified for sterilization to make sure we weren't introducing any microbes into the environment of the bubble. And you'll notice we didn't do a pre-intervention endurance test here because exposing them to a treadmill would contaminate them, and we wouldn't be able to keep them sterile. So we needed to very carefully track voluntary running wheel behavior to be able to compare training responses. So that's why we needed these telemetered wheels. So here's the results of the antibiotic treatment on wheel running behavior in these animals. You'll notice that the red, the animals given antibiotics, show a reduced running volume relative to the control animals. However, while reduced, it was not statistically significantly lower. There was a P of for ABX of 0.11 and ABX by time interaction of 0.11. So it wasn't statistically significant. So in order to kind of prove that maybe there's a reduction, we might need a few more animals here in order to do that. In the germ-free mice, we actually did achieve statistical significance in the interactive term and almost in the main effect term. So you can see here in blue, there was lower running in the germ-free mice relative to these control animals. It really wasn't lower in the first few days, but after about 5 days, the animals in the germ-free group leveled off at a level that was significantly lower in some instances than the control group, at about 10 kilometers per day versus maybe about 15 over the course of the study. We're actually in the process of looking at circadian rhythms. There's some data that demonstrates that germ-free mice have differences in circadian rhythms, and we'll see if that bears out with running wheel. Nobody's ever done a running wheel study in germ-free mice to my knowledge. If we express the data as cumulative training volume, you can see here that there's the graphs before kind of bear out in this graph. There's a significant reduction in cumulative volume of the germ-free animals, about 74% of the load that the control animals got. And then the antibiotic group is about 78%, excuse me. So based on this, we would predict that any adaptations to exercise might follow this kind of scheme down here on the right, where the control animals are greater than the antibiotic treated animals that are about equal to the germ-free animals. But I remind you that both the antibiotic and germ-free animals still had a pretty good stimulus. They still ran quite a bit. We didn't see any effect on final body weights across the treatments, but we did see some effects on gonadal fat pad weight, perigonadal fat pad weights. Voluntary wheel running reduced perigonadal fat pad weights, and antibiotics alone actually reduced perigonadal fat pad weights. And there was actually a trend for additive effect of antibiotics and wheel running shown here, but it was only about 0.07 was the p-value. Interestingly, the germ-free mice had lower perigonadal fat pad weights, and exercise had little effect on those fat pad weights. When we looked at fasting blood glucose, there was no effect of wheel running, probably because these are healthy, mice anyways, healthy young mice. But antibiotic treatment did reduce glucose levels. And there was no effect of wheel running in the germ-free mice as well. When we look at the treadmill performance test, so on the left, you can see time to fatigue in minutes, and on the bottom, distance run in meters. You can see we did pre-testing in these animals, so there's no difference before the wheel introduction, but after the wheel introduction, after 6 weeks, the animals that were exposed to the wheel had significant increases in treadmill performance. And importantly, antibiotics did not have any effect on that. So the antibiotics did not block the adaptive response, at least in terms of performance. In the germ-free mice, a bit different and interesting situation. If you'll notice, I've tried to keep the scales of these graphs the same. And what you will notice that even germ-free mice that are sedentary don't run very well on the treadmill. So you can see here, a control animal with microbes runs 45 to 50 minutes, and these animals that don't have microbes only run about 15 or 20 minutes. There was a statistically significant increase in the animals that were exposed to the wheels. But the magnitude of change relative to the change in the animals containing microbes was definitely lower. So they did not adapt as well. So there was a threefold difference in endurance, threefold reduction in endurance in the germ-free mice. And the germ-free mice did improve their response, but not to the extent that conventional mice did. So lastly, we took gastroc and soleus muscles.
1 minute, Jeff.
Thank you.
1 minute.
Yep. So lastly, we took gastroc and soleus muscles, and we've done some preliminary gene expression analysis, mainly on the gastroc. And this is kind of where I need some help from you. We also took fecal samples for microbiome and metabolome, but we haven't gotten to that yet. So we did PGC1 alpha, mRNA expression, master regulator mitochondrial biogenesis, and not surprisingly, wheel running increased that. But antibiotics had no effect on that increase. What was surprising was that the germ-free animals expressed very, very high levels of PGC1 alpha, and I have not been able to find this in the literature. So this might be a novel finding. And of course, another interesting finding is that the wheel running did not affect PGC1 alpha in these germ-free mice. We looked at PPAR gamma, no real exercise effects, but high levels of expression in the germ-free animals in the gastroc muscle. We looked at GLUT4. Exercise actually decreased GLUT4 mRNA relative to the control. Germ-free have low levels of GLUT4 mRNA expression. GLUT4 evidently is post-transcriptionally modified in muscle in response to training to increase protein levels. But we did not detect any differences. In fact, we saw a reduction in GLUT4 mRNA. MCT1 was reduced in the gastroc of the germ-free, and MCT4 was actually reduced in the gastroc in response to wheel running, not affected by antibiotics, and germ-free was fairly similar to the antibiotic to the control mice. So the take-home messages are that endurance exercise independently affects the gut microbiome of mice and humans, changes who's there, changes what they're doing. Transplant of a gut microbiota from an exercise donor to a sedentary recipient improves colon health. Broad spectrum antibiotic treatment during endurance training does not appear to affect wheel running significantly, at least, or endurance training adaptations, certainly. And germ-free mice exhibit reduced wheel running behavior, endurance exercise performance, and endurance training adaptations. So with that, I want to thank Jacob, Lucy, Noah, and Selena, the main drivers of the data, and many others. Our funding. I want to thank you for listening. And if we don't have time for many questions or comments, please send them to me at woods1@illinois.edu. Again, I'm really looking for some feedback on what next to look in the soleus and the gastroc muscle in particular. Thank you.
Excellent. Thank you very much, Dr. Woods. We've already got 3 questions, and I think we have time for probably 2. So the first question is, Dr. Woods, in your opinion, can a child of endurance athletes or exercised persons inherit an improved microbiota?
So yeah, so you obviously you do get microbes when you're born from your mother. So yes, and of course you're all living in the same environment, hopefully, right? So the environment that you live in has a big driver on your microbiome. The food you eat. So yes, I would say that people who live in families or in close proximity tend to have microbiomes that are similar, but over time they differ because you go out in the world and do different things, and like I said, there's high inter-individual variability in people's microbiomes.
Okay, thank you. Avery Hinks has asked, great presentation. Is there any similar research out there on microbial adaptations with resistance training rather than endurance training?
Excellent question. Very few. So this is a hot area where you can look at. I think maybe some work out of Roger Fielding's lab with aging, and I think Michael Lustgarten. They work together up in Boston. So they've done a little bit of work on that in that regard, but very few studies with resistance training, much more so with endurance training.
Okay, we actually have time for the 3rd question. Anthony Shime here at York. Were the germ-free mice the same strain as controls? Also, in the obesity study, might altering the diet of the obese participants account for the results?
So yes, the strain was C57 Black 6. We made sure of that. So the second question was,
sorry,
was the diet different in the obese subjects?
If you altered the diet intentionally of the obese participants, also in the obesity study, might altering the diet of the obese participants account?
Yeah. So the way we did that was we took their a few dietary records from them, and then we fashioned a meal menu right before the fecal sample that reflected their normal diet. And really what we wanted to do is make sure they ate the same things for 48 hours or so before each fecal sample, pre-intervention, post-intervention, and then 6 weeks after the washout. And our analysis, reflects that. And so they really didn't change the types of foods they eat in those acute diet manipulations. But we wanted to make sure they were exactly the same. And a lot of studies don't go to that length to do that. And there can be acute dietary induced changes in your microbiome and metabolome. So, many studies should try to do that. Short of putting them in a metabolic ward and feeding them ourselves, I think we did a pretty good job of controlling for the influence of diet.
Excellent. Well, that's fantastic. Thanks a lot, Jeff, for coming today and presenting, and great round of Q&A. Extremely unique topic, so we're all very fortunate you did that. Thanks very much.
Thanks, Chris. And if anybody's got any suggestions for where we need to go with this, you muscle experts, woods1@illinois.edu.
There we go. Take care. Thank you.
Yeah.